How is cholinergic function measured in schizophrenia research? Have you ever wondered how scientists study the cholinergic system in schizophrenia? Understanding this can help us grasp how new treatments are being developed. Cholinergic function is measured using several methods that look at different parts of the cholinergic system. These methods include measuring neurotransmitter releases, receptor availability, brain structure, and cognitive performance linked to cholinergic activity. First, researchers focus on neurochemical measurements, especially on acetylcholine releases and activity. Techniques like microdialysis allow scientists to sample the extracellular fluid in specific brain areas. This helps them measure the levels of acetylcholine released over time, giving a picture of baseline cholinergic activity. However, microdialysis has its limits. It cannot differentiate between various cholinergic neuron populations or capture rapid bursts of acetylcholine releases. For those quick events, amperometric recordings come into play. These recordings provide high temporal resolution to detect brief bursts of acetylcholine releases, which are crucial for cognitive processes. Next, neuroimaging techniques, such as structural magnetic resonance imaging, are used to assess the integrity of cholinergic brain structures. One key area of focus is the basal forebrain cholinergic nuclei, which provide cholinergic input to the cortex. Studies have shown that reduced volumes or gray matter integrity in this area are linked to attentional deficits in schizophrenia. This suggests that structural changes in the cholinergic system may be related to cognitive symptoms. Another important method involves receptor-level assessments. Researchers measure the availability and density of cholinergic receptors in the brain. Post-mortem studies and in vivo neuroimaging have shown widespread reductions in muscarinic and nicotinic receptor levels in individuals with schizophrenia. These receptor deficits often correlate with the severity of symptoms, including cognitive impairments. Cognitive testing is also a practical way to measure cholinergic function. Tasks that are sensitive to cholinergic modulation, like attention tests, help researchers correlate cognitive performance with the integrity of the cholinergic system. Deficits in these tasks often indicate underlying cholinergic dysfunction in schizophrenia patients. Lastly, molecular and biochemical assays are used to measure enzymes and transporters related to cholinergic signaling. For example, choline acetyltransferase is responsible for synthesizing acetylcholine. Altered activity of this enzyme in schizophrenia supports the idea of cholinergic dysregulation. Together, these methods provide a well-rounded assessment of cholinergic function in schizophrenia. By examining neurotransmitter dynamics, receptor status, brain structure, and cognitive outcomes, researchers can better understand how the cholinergic system contributes to schizophrenia symptoms. This knowledge is essential for developing new cholinergic-based treatments that target cognitive and other deficits associated with the disorder.